So I also met my wife at that point, who was, stood by me throughout all the years and through the clinical training. And it was in the early 1980s, we'd returned to Baltimore. I was working on red blood cell problems. I was a hematologist at Johns Hopkins as a trainee, not a faculty member. Very happy, a low salary, riding my bicycle to the lab every day. And tragedy struck. We had two beautiful little daughters, and Mary was pregnant for the third time. And late in the pregnancy, she had a spontaneous, early, premature labor, and led to a birth of a very severely affected little girl who had a profound form of cerebral palsy. And it was a wake-up call, I think, to me. In part, all these things we're working on in the laboratory seem so interesting, but what do they do in terms of the human lives of individuals that are affected by these problems? And we, of course, all know these problems, and our little girl eventually died. It turns out, of course, this is not so rare. In the late 20th century in the United States, about 1% of children born would not live to their first birthday. And of course, in the less advantaged American communities, in the African American community, it's about twice that. So first off, I needed a job. And fortunately, Victor McCusick, the chairman of medicine at Johns Hopkins, offered me a faculty position. And I thought I would focus our work on diseases affecting the newborn. Not that we could change anything, our daughter was already gone, but it caused us to work on, our lab to work on problems of the red blood cell at the time of birth. And of course, the Rh antigen is a well-known clinical problem affecting neonates. And so we were working on that, and we made a completely serendipitous observation. By purifying the Rh antigen for the first time, we isolated a second protein, a protein that we didn't expect to see. No one had seen it before. And we didn't have a clue what it did, but it was extremely abundant. And we got some protein sequence from this, and we found that it had related proteins that had been identified. A protein from the lens of eye, a protein from bacteria, proteins from plant tissues. And I think, like scientists, you're very curious when you discover something new, but the importance is to explain what it does. And we were stuck. But by being kind of generally, I think, friendly, eager to talk to other scientists, I got some very good observations. And it was, in fact, a discussion I'd had with John Parker at the University of North Carolina, that suddenly the lights went on. He said, red cells, the protein was also present in renal tubules, cousins in plant tissues. He said, Peter, have you considered this might be the long-sought water channel? And of course, it never occurred to me water channel. But for over 100 years, physiologists had questioned, how is it that some tissues are so freely permeated by water and others are not? How is it we can regulate, at times, water permeability? Anybody sitting in the center sections by the end of this program, if you had a large cup of coffee, you're going to feel a fullness in your bladder. And this represents one form of the need for a plumbing system in biology, a protein that allows water to enter and leave cells in a rapid and a regulated manner. This is really what allows osmosis to occur in biological systems. And in fact, that turned out to be the case. So the eureka moment was to test this hypothesis in a simple experiment. I won't go through the details, but it worked. And it really did change things in terms of our laboratory. Instead of being a small, and we were a very small laboratory, there was suddenly a large amount of interest. There was a lot of ramifications. Studies were undertaken to explain the movement of fluids in the eye, which may be involved brain, which may be a problem in brain edema, in pulmonary fluids, which those of us out hiking are going to have some experiences with here out at high altitude, and renal concentration. So I think it's an example of a chance observation, a serendipitous observation, with the very, I think, important insight of other scientists, some good luck, and then a very important factor, and that's federal funding for science. Because we were and remain funded by the federal government. Your tax dollars, part of these go to the National Institutes of Health, and through the National Heart, Lung, and Blood Institute and other institutes, we were able to be funded all these years. So I'm probably going on too long. I'll just briefly, in closing, explain a few things and maybe take a little bit of the aura off the Nobel Prize. The work had gotten a lot of attention, and it was clear that I was being invited repeatedly to Sweden to give some special lectures. Now, all scientists probably have this little daydream about Sweden, the Nobel Prize. And I think the challenge is not to take that too seriously, because there's a lot of great science. But I remember distinctly, I think it was about 1999, when I was invited to give a special lecture at the Karolinska Institute, the medical school in Stockholm where the Nobel Prize in Medicine is decided. I flew all night, and as I would always do, I'd fly on the cheapest seats on the airliner, the economy minus section. And I remember arriving early in the morning at the Arlanda airport and getting into the queue to go through the pass control. And of course, the business class, they got through first, so a lot of gentlemen in suits, well-dressed with briefcases, and the pass control officer would ask each the purpose of his visit to Sweden, and they would respond, business. Next. Purpose, business, next. And he came to me, and the officer looked at me, what is the purpose of your visit to Sweden? And I said, well, I'm here to give a special research lecture at the Karolinska Institute. And he looked at me, he said, are you nervous? So I said, well, yes, in fact, I am nervous. And he looked at me again, he said, well, you should be. So it was a few years later, early in the morning, the telephone rings, and the voice on the other end announces that this is an important telephone call from Stockholm for Professor Peter Agri. Are you Professor Agri? I said, I sure am. And he told me I would share the Nobel Prize in Chemistry with Roderick McKinnon. And it's interesting, I had not worked with Rod. Rod worked on ion channels, how salts cross cell membranes. We worked on how water crosses cell membranes. And of course, tears and sweat and urine, these are salt solutions, mostly water. And I think, but Rod being someone I've always greatly respected, it gave me some reassurance that we have some credibility here to share the prize with Rod. And so as I sprinted down to have a shower, my wife, Mary, called, and we've been together all these years. She's always been a great support. She called my mother back in Minnesota. Dad had died 8 years earlier, but mother was out there living alone. And again, this is a farm girl with no formal education. And she explained that her son would share the Nobel Prize in Chemistry. And mother thought for a moment. She said, Mary, tell Peter not to let this go to his head. Good advice. And of course, that was the Washington Post, the headline, Nobel Prize winner's mother. So I came to work that day, and of course, life had changed, the big celebration in the laboratory. The most amazing thing, the university president came over to see me. I had no idea we were such close friends. And I went to the voicemail where there were lots of requests for interviews in the Lehrer NewsHour, New York Times, and I came to a familiar voice, one of my colleagues from Norway, with whom we'd worked on the neuroscience of aquaporins and brain edema. And he was so excited, the voice said, Peter, we've just heard the news. It's unbelievable. And then there was a pause, and the voice came on and said, oh, no, it's believable. And as I drove home that evening, I saw that the marquee in front of the discount liquor store at the local shopping center had a big sign that said, congrats, Dr. Agri, as if I was their best customer or something. So life has changed in some ways. I think it's provided a bully pulpit to take science to the public more. We've had a lot of opportunities to lecture at universities and to the public, right? To give technical lectures about aquaporins in health, and in agriculture. The roots of plants have these proteins. But I also like to speak to the public because I think the public are the great supporters of science. And I think by communicating the importance of science and the joy of science, it's demystified to some extent, but I think it's a very important thing to do. And I'd never dreamed I'd be invited to be on the David Letterman show. I actually declined the invitation. They wanted me to assemble IKEA furniture. I think the joke was that here's a Nobel Prize, he can't do it, but our whole house is IKEA furniture. But it coincided with an invitation to go to the Nobel Peace Symposium in Minnesota where Jimmy Carter was speaking, which I felt was much more important. But I was on the Colbert Report not too long back. Gil Oman can tell us about that. But I think it does provide an opportunity to speak to the public because I think science is what often drives the discoveries that leads to the innovations. And the innovations are, I think, in many ways what underpin the economy on which our country is based. So I think science has been good to me. I plan now to continue our work. We're working on malaria, our work at Duke University, which is a lot of fun, the MD PhD program. And I think if we continue to invest in science and encourage young people to take an interest in science, good things will happen. Well, so with that, let me thank you again for the invitation to be here.